Okay, so hopefully that was enough time to get through the warm-up. I appreciate everybody being patient with me. Again, we're still trying to get through the kinks of teaching both virtually and in person simultaneously. Today is not the 26th. I changed the date. Today is March 1st, not February 26th, 2021. And it's the fourth day of our fifth unit. Today's lesson is going to be about this process of translation. So I do want to just ask, and I see all four of the people who I have here in person with me have their notebooks out. They are ready to go. They finished their warm-up. They've got their Chromebooks out as well. So they are four students who are prepared to succeed in this lesson. I want to make sure those of you who are at home are following those same practices. All right, so it's always a good idea to try to write things down. Literally, research shows that when we're writing things down, it helps us to memorize it. So more so than just typing, more so than just listening, more so than just watching, actually writing down notes is a good idea. And it's always a good idea to start on this lesson page, this title page. So we've got an objective today that states that biologists will be able to explain how DNA and RNA code for proteins and determine traits. And we've got an essential question that asks, how does genetic information code for the production of a protein? And it's made out of amino acids. So a few things to mention before we get started. Number one, I got like 2 hours of sleep because of a fire alarm going off. So I apologize if I sound lethargic, but I'm going to need you guys to jump in and help me out, keep me energized throughout this lesson. Number two, we do have a midterm exam approaching. In this class, I believe it will be a week from today on Monday, March 8th. So please just be aware of that and be ready to study. Throughout this week, we will be doing some review, and we're going to try some new things with breakout rooms to really focus in on some standards that we've struggled with so far. But I want you guys to be actively engaged in that process as well. And then lastly, what was the last thing I was going to mention? March 1st represents the beginning of National Women's History Month. So we will be talking about some of that history, and especially women in science, but also just thinking about the role of some prominent women in society. So keep that in mind, and hopefully you all have an opportunity to talk about this National Month in your other classes. If not, feel free to be the leader and remind your teacher and be like, hey, March is National Women's Month. So we should be discussing some things related to the contributions of women to our society and to societies across the world this month. So don't be afraid to step up. And as a teacher, I would say that would be very, very impressive if a student did that. Any questions before we get started? Yes, ma'am. A pen? Yeah, you can just grab. There's one right there on the table. Do you see it? And you can keep it too. Ooh, okay. Christian is here. Thank you for joining us, Christian. I appreciate it. All right. So we talked about this on Friday, but the process of transcription takes the DNA message and transcribes it into a similar mRNA message, but there's an important difference, several important differences. One of the most important differences is that RNA has a U, whereas DNA has a T. I don't remember if we did this exact strand on Friday. We may have. But let's see if we can transcribe this complementary DNA here. So everywhere I see an A in DNA, it should become a U in mRNA. Everywhere I see a T, it should become an A. Everywhere I see a G, it should become a C. And everywhere I see a C, it should become a G. So who thinks they can kind of knock out this strand for us? What's the first letter? And I kind of have that written on the board here too, for those of you who can see this. What's going to be the first letter in the mRNA sequence? Are we confused? Yeah, you guys can answer too. Okay, good. And I saw you. So we're transcribing this. A is going to become U. Everywhere I see an A, there's going to be a U. What's going to come next, man? Good. Everywhere I see a T, this is what we call transcribing. How do you feel about that, Ayana? You feel good? Okay. Alex, how do you feel about it? Okay. We'll keep practicing with it. And then for the folks at home, I know you guys are all on mute, but hopefully we're understanding this concept. Breon is helping us out here. He's telling us that when we see an A gets transcribed to U. When we see a T is transcribed to an A. So again, this next letter is an A, so we have another, what do we have? Another U, good. And then what? Another A, good. Who thinks someone else can pick up these next few letters? What does the G become? Good. Thank you, Ayana. The G is going to become a C, then what? Then G. Yeah, G and C are always, they're pretty easy just because they always go together. So even if we're talking about just figuring out the complementary strand of DNA, we're always going to end up with C and G going together. Okay, what comes after that? Yep, T becomes an A, then CG, UAA, good. UC, good. Excellent. All right, so we got some help there, a lot of help from Breon and some help from Ayana as well. But if you folks at home are struggling at all, please just let me know and we can make sure that we cover it and I'll try to explain it in a different way. As I said, you can kind of see it over here on the board. Please make sure that you are at least writing down the bases that are complementary to one another. If we've got a strand of DNA and it's becoming a strand of RNA, these are the rules we should follow. Okay, cool. We good to go? Alex, you good? Ayana, you're good? Okay, cool. Okay, so last Friday, we focused on that very first process of transcription. Today, we're moving on in this overarching process of protein synthesis to discuss what's called translation. So transcription is moving from DNA to what is called messenger RNA or mRNA. Today, we're going to be moving from that mRNA to an amino acid. And hopefully we remember from way back in Unit 1 in January that amino acids are the monomers of proteins. Okay, so again, remember, genes code for the proteins that our cells make, meaning that they have the instructions. They are the instruction manual to eventually make a protein. So we're moving on to the second step today, translation. So the goal, I know there looks like a lot of information here. At the very least, try to write down what is highlighted in yellow, and we can take our time. I'll stay on this slide for a long time. But the goal of translation is to read that mRNA sequence and then use it to begin linking amino acids together to make a protein. This process occurs in ribosomes. Ribosomes are the organelles that are responsible for making proteins. I try to remember the order of these two processes because transcription is alphabetically before translation. So therefore, transcription is the first process, followed by translation. So while folks are starting to wrap up their notes, I have a few questions. As I said, amino acids are the monomers of proteins. Does anybody remember what the monomer of a carbohydrate is called? Folks at home, please feel free to jump in here. What's the monomer of a carbohydrate? I love what I see folks doing. They're looking back in their notes. That is an excellent habit to be in. Good. Ayana got it. The monomer of a carbohydrate is called glucose. What about the monomer of a nucleic acid? What's the monomer of a nucleic acid called? Wonderful, Brian. Yeah, that's called a nucleotide. So we're going to be doing more. We're going to be more intentional about our review this week. I'm just going to kind of hit you guys with some questions as we go, because we've got to be mindful of our midterm coming up. Anybody need more time here? Okay. Good, Brian? Okay. All right, so let's see. We're going to go over these steps a little bit more. I'm going to go back. Just give me a second. All right. Actually, we'll take some time to write this down. You don't need to write down everything, but like I said, at the very minimum, please write down what is highlighted in yellow. I know this is a lot, but there won't be much more writing after this slide. So just bear with me here. And I'll give you all plenty of time. The first step, the ribosome attaches to the mRNA strand, and this happens in the cytoplasm of the cell. So Remember when we were talking about transcription on Friday, in a eukaryotic cell, that happened in the nucleus. Now we've moved out of the nucleus and we've moved into the cytoplasm. That's an important difference. The ribosome is going to read the mRNA in groups of 3 bases. So it might be AUG, UGC, CUA, so on and so forth. We call those groups of 3 bases codons. Each codon, as we will see later in the lesson today, corresponds with a specific amino acid, or it could even tell the ribosome to stop, which signals the end of translation. We'll talk about that a little bit later on as well. The third step, a tRNA molecule with the correct anticodon to match the codon brings a specific amino acid to the ribosome. And the last step, as the amino acids are brought, they get linked together to form a polypeptide chain, which will eventually fold into a protein. So I'm going to give you all plenty of time to write this, to soak it in, to read over a few times, and then we'll move on. Okay, so I just want to read through these steps one more time as folks start to finish up writing. And I want you guys to just soak this information in. It seems like a lot, but as we start to see what it looks like in images and videos, it will make more sense. First up, a ribosome is going to attach to that mRNA strand in the cell's cytoplasm. So keep in mind that the mRNA strand was just made via the first process, transcription. The second step here in this process of translation, the ribosome reads that mRNA in groups of three, and we call those groups of three codons. So it could be UUU, it could be AAA, it could be UAU, any group of three. We're going to read the ribosome is going to read those groups of three and then use those messages in the third step to bring a specific amino acid to the ribosome. And then lastly, those amino acids get linked together. They get bonded together. And in the process, they create what is called a polypeptide chain, and that chain will eventually become a protein. So let's take a look at how this works. Phone is my remote here. All right. So some important structures to note here. We see the mRNA strand down there at the bottom. The ribosome is that big blue thing that's kind of in the background of everything. So this entire process of translation is happening inside of the ribosomes. The first step, as we noted, is that the ribosome is going to attach to the mRNA. Then it's going to start to read the mRNA. And it reads the mRNA in groups of three called ribosomes. I'm sorry, called codons. That's my 2 hours of sleep coming out. The ribosomes are going to read the mRNA in groups of three called codons. So we can see a codon there in the example GAU. The third step, the tRNA, which is a type of RNA that literally looks like a T. In this case, they're kind of like the golden yellow thing. The tRNA with the correct anticodon brings its amino acid to the ribosome. The anticodon is not something we really need to worry too much about in this class. As long as you know what the role of the tRNA is, you'll be good. And we're going to talk about how we know which amino acid is going to be brought in a couple of slides. And then lastly, the fourth step, we can see that the chain of polypeptides is beginning to form. So each of those colorful circles is an amino acid. As we link them together, they start to form a protein. So this might be something that you take a picture of, or that maybe you might want to come back to and try to redraw in your own notes later on. As you all know, I'm a big fan of graphic images and mind maps. So we've got some really important structures here. Almost everything is going to be important for you to know, except for the peptide bond. That's chemistry. And then the anticodon is not super important in this class, but everything else you should be familiar with. Any questions up until this point? No questions? You good, Adrian? Alex, you're good? Okay, cool. Okay, so during translation, the cell must change the language from nucleic acids into amino acids. And this is like translating to different languages. So we've got some Hebrew on the screen. And in English, this would say, "You are the most amazing students ever." So we're literally going from one language to another. In this case, in the process of protein synthesis, we're going from the language of nucleotides to the language of amino acids. So instead of A's and T's and C's and G's and U's, now we're going into another type of language called amino acids. Okay, cool. So now that we have seen or we've learned a little bit about translation, let's watch this video. We're going to see both transcription and translation happening here. And then we'll talk more about the process of linking the amino acids together.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
What I want you all to think about is the fact that this is a very, very fast process from start to end. In a bacterial cell, this takes about 1/10 of a second. And so our cells, and we've got trillions of cells in our bodies, they're doing this process constantly, and it's happening extremely, extremely quickly. So it's really, really kind of, a beautiful natural process, but one that we don't think about very much, but it literally makes us who we are. Okay, so this is where it's going to start to get a little interesting. I just want you guys to bear with me. Those of you who are here in person, I passed out a laminated piece of paper. We are going to use that. And you should see, if you flip on the other side, you should basically see this same chart that we have on the screen now. Okay. So in nature, there are 20 amino acids. And again, the amino acid is the monomer of a protein. These amino acids have different chemical properties. They have slightly different shapes and slightly different sizes. And so when we combine them in different combinations, we end up with proteins that can do different things. But as the ribosome is reading those groups of 3 bases, aka a codon, as the ribosome reads these codons, Each one of the codons is going to tell the ribosome, hey, I need this amino acid to come next in the order. There are a few codons that don't code for an amino acid. They just code for a stop message. They tell the ribosome, okay, that's the end of translation. We have successfully made the correct protein. You can cut it off now. And we can see the two of those three, and there should be a third one, so my apologies. But two of those three codons are on the screen, UAA, UAG, and the last one is UGA. Okay, we good here? Cool. So let's talk about how to use these codon charts. It seems a little complicated, but I promise you it's not. On the left side of the codon chart, and I know you all at home don't have a physical copy in front of you, but hopefully you can see it on the screen. On the left side of the codon chart, you can find the very first base. Let me get a copy for myself so I know the in-person students. Okay, so on the left side of this codon chart, you can see it says first base. For my students who are in person, it says first letter. So again, in these groups of three, obviously there's always going to be one letter that comes first. Once you identify that first letter, you now are only looking in that row, in that letter's row. So we'll see what that looks like. Then there's a second letter or a second base that is on the top. So again, once you identify what the second base is, now you're only looking in a specific column. And then the third base is on the right. And once you know what that third base is, you should be able to, at that point, identify the amino acid. But let's take a look at exactly how to do this. And there is another codon chart as well that might be easier for some of you. It's just a matter of preference, but we'll talk about that as well. And before we move on, I just want to mention that this is why the sequence of the mRNA matters so much. Because, for example, UUC is not the same thing as UCU. Even though we've only switched around two letters, that's going to give us a totally different amino acid. So this is why the order is so important, because we're going first, then second, then third. So just be aware of that. All right, so let's focus on this codon chart on the left first. It's a matter of preference which one you use. I think when I was in high school, I learned how to use the chart on the left, but I think a lot of my students at West Meck so far have preferred the chart on the right. So let's just take a look at both of them. So for example, let's say we've got the codon GAC. What's my first letter there? What's the first letter of that codon? Don't think about it too hard. What's the very first letter here? Good. The first base or the first letter of the codon is G. So once we've identified that G is the first base, now we should limit what we're looking at. We know that G is the first base or the first letter. So all we need to look at is the horizontal row of that very first base. Only that row, which I have now highlighted in red on the screen. And now we don't need to pay attention to anything else because G is our first. The second base is A. So now we're looking at the top where it says second base, or for my students in person, your physical copy says second letter. We're going to look at the second letter, and it is A in this case. So now we're only looking in that column. Now, keep in mind, and I know I wasn't, you guys probably couldn't hear me at home. We're looking at the column for the second letter, which is A, but keep in mind, we can only look at the overlap of that red row and the green column. We can only look at the overlap there. Everything else has already been eliminated. We can't look at, for example, PHE in the top left because that requires that U would have been the first letter. We can't look at LEU in that second column because that requires that C would have been the first letter. We know that the first letter was G. So we can only look in those, in that G column, I'm sorry, in that G row down there. So we're only looking at the overlap of the row and the column. So now we're limited to only two options. What are the two possible amino acids now? You can just give me their abbreviations. What are the only two possible amino acids? Good. The only two possible amino acids are ASP and GLU. And then our third letter, of course, is C, which is blue here. So identify the third base of the codon. In this example, we've got C, and then we find the amino acid for that letter. So GAC gives us the amino acid ASP, which is an abbreviation for a longer word, but you all don't really need to know that. You don't need to memorize this chart. You don't need to memorize all 20 amino acids. You just need to know how to use the chart. We'll get more practice with it. Don't worry. Any questions up until this point, though? Brian, you're good. Alex, how do you feel? Cool. All right. Let's talk about the other one, though, and I think some of you might prefer this one. So just to show you that these charts work the same, and for those of you who are in person, you can flip it over and you'll see that there's this circular chart there. But just so you can see that these charts work in the same way, we should get the same answer. Remember, the answer we got here was ASP. So if we do everything correctly, we should end up with that very same amino acid. So our first letter is G. The way this works is I'm going to start in the middle of the circle. We've identified the first base of the codon, which is G. Starting from the middle of the circle, I'm going to find that base's section of the pie, for lack of a better term. And then I'm going to just literally ignore every other section. So once I identify that the top left quarter of the pie is for G, I can ignore everything else. So there's G, and everything else literally gets blacked out. In my mind, I don't need to pay attention to it. So now we see that we've limited our options pretty substantially. Now we only have 5 possible amino acids already. Now we're going to move outward. So you start in the middle of the circle and you start to inch your way out from there. So the second base of the codon is A. Now I'm only paying attention to the section of the pi for the second letter being A. It looks like that. So again, everything else gets blacked out. So now we can see I've got G as my first letter. Then I've got A as my second letter, and I'm moving outward. I start in the middle of the circle and I move outward. My last letter, of course, is C. So again, now I move outward one more step and I see where C is located in the pi, and I'm only paying attention to that section of the pi. And that tells me what the amino acid is. Again, we've got aspartic acid that is ASP. So we did it correctly. Which one do you feel like you prefer to use? This one? Yeah, I think this one, typically students do prefer this one. And, you know, for those of you who are here in person, I can even print one of these out and you can just use, you know, you can mark it up however you want. So you can identify your amino acids. So let's get some more practice with this. I'm going to try to use both charts, but use whichever one you prefer. Okay. So keep in mind, this is only for mRNA. That's why we have U's. We don't have T's. This is not about DNA. We've already gone through this process of transcription, so we already have the mRNA. So our first example here, I want the mRNA codon AGU. My first letter is A. I do it like this. Let's see. Yeah, I'm only paying attention to this section here. So now I could be talking about the thymine, asparagine, lysine, blah, blah, blah. But I know that my second letter is G, so I know I need to be in this section. The overlap. So it can only be two things at this point. Serine or arginine. These get ruled out because the second letter is G. But we know the second letter is G, so it's got to be over here. And then my last letter is U. My third letter right here. So the amino acid is serine. I do another one. Okay. GAC, I'm going to do another one by myself here just to demonstrate. G is the first letter. So I'm only looking at that very bottom row. I'm going to highlight it in purple here. Oops. Okay, I skipped some steps here. G was my first letter. I only look in the bottom row. A is my second letter, so I'm only looking in that third column in the bottom row. So I only had two options at that point, aspartate or glutamate. But then I look at my third letter, which is C, and that tells me it needed to be aspartate. Okay. Let's do one together, essentially. You don't need to use this chart, but just walk me through how you would do it. And this can go for anybody at home as well, but my people in person, it'll be easier since I'll be able to actually see what you're talking about. So how would you attack this? How do you figure it out? GAG is the codon. First base is C, good. Oh my gosh. Okay, I'm sorry, Brian, what did you say? Yeah.
Good morning, Wittenberg High School, second block teachers, please. If your students are not aware where they need to go to for home time, Please pull up the assignment document and show it to your location. This is our location B. For many students, this is their first day on campus, and they may not know their home assignments. Again, here's these last few minutes to remind students of their location, whether they're going to a literacy cafeteria or to their home teacher. Thank you.
Yes. And so CAG, we're only looking here. The abbreviation is GIU. You're right. Isn't it? I'm pretty sure it's GIU. Yeah. Good job. Okay, another one, AUC. Someone walk me through how they might figure out the amino acid for AUC. My folks at home, you all have been really quiet today. No, there aren't any messages in the chat. So if one of you thinks you can do AUC, please shout it out. Go ahead, Brian. Okay, let's see. So okay, good. You're feeling confident about it now. So we did A as the first letter. U is the second letter. So now we're only looking in that kind of the third row, first column. So that limited us to ISO, which is isoleucine, or MET, which is methionine. To figure out which of those two we're talking about, we need to look at the third letter of the codon, which is C. And because we're talking about AUC, C is that third letter, then the correct amino acid is isoleucine. Good job. All right, let's try one with this chart. And maybe Ayana or Ardeja, one of you can kind of walk me through this. So we've got AGU. Good. So we're on 2 at this point for amino acids. Okay. Good. And then which one is it? And how did you know that? Good. So we started from the middle and you moved backwards. Look at A, second letter is G, third letter is U, so that tells us there's. Good job. So we're starting from the middle here. I know you guys probably didn't hear that, but Ardeja was able to walk us through it. We start in the middle. From there, we look at the first letter, which is A. It tells us that we're only paying attention really to the bottom left of this chart. Then from there, the second letter is G. So we can ignore that portion of the pie that says A. We can ignore the portion that says C. We can even ignore the portion that says U. We're only looking at G. So Ardeja was able to tell us at that point that that limited us to only 2 options, ARG and SER. But then she was able to figure out which of the 2 that we're talking about because she knew that the third letter was U. So AGU gives us the amino acid serine. Excellent job. Okay. GAC. You think you can do that one? Go ahead. Yeah, whichever you prefer. Good. Yep. Good. C is the last letter. Wonderful. Yeah. And see, guys, you get the same answer whichever one you prefer. So whichever one you're more comfortable using, I'm cool with. So in this case, Ayana chose to use the kind of the more chart-like codon chart, the more table-like codon chart. And we had the codon GAC. So she correctly identified G was our very first letter. So we only look in the bottom row of that table. A was our second letter. So now we're looking in the bottom row still, but this time we're only looking in the third column of the bottom row. And then our last letter is C. So she was able to tell us that the amino acid is aspartate. Good. Okay, so let's put it all together now. Let's demonstrate the processes, two processes of both transcription and translation. So the first thing we want to do here is transcribe from DNA to mRNA. So what does TAC get transcribed to? It becomes U. Because it's mRNA. So we don't have any DNA. So A becomes U, then C becomes G. All right. So again, for my people at home, I'm sorry. It's difficult to hear when I'm away from the computer. But what we're doing here first is transcribing. We're going from DNA to mRNA. So T is transcribed to A is transcribed to U, C is transcribed to G. We can see those rules over here, right over there. What's that next codon going to be? GGC. What does that get transcribed to? CCG. What about the next codon? Good. You guys are getting fast with it. AUC. And the last one? UAA. Wonderful. Okay, now this one's going to take a little bit more time. We've transcribed everything correctly. We went from DNA to mRNA. That's transcription. To go from mRNA now to amino acids is translation. So you need to use this codon chart to do that. Can anybody tell me what, which one? Good, wonderful. All right, so that first codon, AUG, if you're using your amino acid chart, we know that that becomes MET, methionine. Now, here's a cheat code, guys. Methionine is almost always going to be first. So just be on the lookout for that. MET is almost always going to be the first amino acid because AUG will almost always be the first codon. So just something to note. Okay, that next one, CCG. What does that become? That becomes PRO, proline, right? Yeah. Good. Then AUC. Yeah, and actually I know it's kind of tricky. The abbreviation is ILE, but you're right. It's isoleucine. And then that last one, what does that give us? Cool. So this doesn't actually give us an amino acid. UAA along with UAG and UGA, those are the three stop codons. So stop is not an amino acid. It's literally telling the ribosome to stop translating. Okay, so at that point, we would be done with whatever protein we're making. So now we've put it together. We've done this process of protein synthesis. We transcribed to mRNA, and then we translated to amino acids. So good. We're going to continue practicing with it, including in today's exit ticket. We've got 9 minutes left. Obviously, I'll still be here. So if anyone, including those of you who are in person, need help with anything, just let me know. But let's go ahead and shift back to Canvas. Yes, sir. Yes, you may. Switch back to Canvas and knock out this exit ticket. If you have any additional time, maybe you'll use that to work on another assignment that you haven't completed. Go ahead. Oh, yeah, it's just right across the hall there. No problem. Okay. Thank you all. For those of you at home, I appreciate it. We'll talk more tomorrow. I hope you have a good Monday. And those of you in person, let me know if you need anything as you navigate the hallways. But enjoy your day. Oh, does anybody need to know where they're going for hawk time? Okay. You are going to room 804 with Miss Dawkins. You know where that is? Okay. See you. Have a good day. What's that? Yes. Do you know where you're going? Yeah, Mr. Reese. That's right upstairs. Do you know who your advisory teacher is? Mr. Sam. Yeah, yeah, Mr. Samick. Why are you not trying to go there? I really don't know because of the protocols that are in place. I think they really want to have students be where they're supposed to be. You can go check his room right now. I don't know, because we have two halls. So one of them is going to be with lunch. So you might be in the cafeteria first, and then you can just talk to whoever. You're welcome. Breon, what's up? The dog. Oh, you're right. Did I?